Good morning everyone. Today we are going to discuss neoplasia. This is continuation of the last two classes. I believe in the last two classes you have learned uh, terminology of neoplasia and metastasis, right? So in today's class we'll be discussing molecular basis of cancer. Let us begin. So cancer is a multifactorial disorder. That means a single factor is not sufficient to lead into cancer. There are several factors that are involved in the development of cancer, like epidemiological factors, like age of the patient. You know, few cancers are more common in childhood, few cancers are more common in elderly people, and the geographic area where you live. Few cancers are more common in certain geological areas, few are more common in some other countries. Like this, epidemiological factors, they play an important role in the development of cancer. And coming to environmental factors. Environmental factors include infectious pathogens from the environment, several viruses, bacteria, whatever may be the organism that plays a crucial role in the development of cancer. Not only that, your food. What is the food that you take? What is the amount of exercise that you do? And your personal habits like smoking, consumption of alcohol, amount of exercise that you do, degree of obesity that you suffer. These are all, they will come under environmental factors. They too play a critical role in the development of cancer. And there are still some unexplained causes are still there. They are now under research umbrella. But apart from these three categories, there are genetic causes. So for the development of cancer, even the genetics play a crucial role. Okay, that is the concept you have to understand. These genetic changes, they can be inherited changes or they can be sporadic also. We all know that few cancers, they run in families. So there is a inherited germline mutation that is being carried out to further generations, and few mutations are sporadic in nature. That means there is occurrence of a genetic mutation just out of blue in a random person. Okay, so understanding the genetic basis of the cancer is what is called as understanding molecular basis of cancer. That is what we are going to do today. Understanding this genetic cause of cancer is what we call it as molecular basis of cancer. Okay, so in molecular basis of cancer, what we are going to do today is we are trying to understand how does a normal cell is developing into a cancer cell, right? So what are the different properties of cancer cells that keep what you call it as that distinguish a normal cell from a cancer cells? That we call it as malignant phenotype of cancer cell. See, a cancer cell definitely has so many qualities that a normal cell doesn't possess, right? That's why that's why it is a transformed cell. That's why it is a dangerous cell, right? So what are the different characteristics of a cancer cell? So that we call those properties as malignant phenotype of the cancer cell. Cancer cells, they possess excessive growth. Their growth is unstoppable. Not only that, they are immortal in nature. And when they are developing in the host, they thrive. They try to survive in the host environment. So they have to protect themselves from the host immune system. So evasion from the host immune response is a special character of cancer cells. Okay, and when they are growing, they can locally invade the surrounding structures so that they will occupy some space. They will nest over there in your body. Okay, and the ability to metastasize, that is detaching from the original site and going into some anatomically not continuous site, and there going and settling there, developing into a secondary. Okay, that is what we call it as metastasis, right? So these are all the different characters that are possessed by a cancer cell so that it stands out from a normal cell. Okay, so let us understand natural history of cancer here. for a moment, right? See, a single cell is transforming into a cancer cell, and it is acquiring all these special characteristics like excessive growth, immortality, evasion from host immune response, invasion, ability to metastasize. These are all the different unique capabilities of a cancer cell, right? How does this cancer cell is acquiring all these special characteristics is one very big question. That is what is the natural history of cancer. Okay, so in this natural history of cancer, if you observe, single mutation, it cannot offer all these characteristics together. Okay, cancer cell obviously it carries so many different mutations. Each mutation is responsible for each of these characteristics. Understand this concept. So a single mutation, it cannot offer complete malignant phenotype. Okay, so somewhere in the development of cancer, at the very beginning of its life cycle, a normal cell has acquired one special character due to the mutations, so that we call that as initiated cell. So this initiated cell, now it has got a potential to develop into a malignancy. The very beginning initiated cell is not a complete malignant cell. It has now attained the capacity to transform into a full-blown malignancy. Okay. So for example, if you take one precursor cell is there, from that a different cell is formed through some mutations. That mutation, it may offer that cancer cell like one of the property, like excessive growth or character of immortality. Only that two characteristics or one characteristic is not enough to transform that cell into a full-blown malignancy. Okay, it has to acquire all the other characteristics also, like it has to protect itself from host immune response, it has to develop the ability to metastasize, it has to develop the ability to survive in a difficult situation. The host cells, they are definitely, they are foreign to this cancer cell. It has to thrive in that new environment, isn't it? So for that purpose, it has to acquire all the possible capabilities so that it can survive in the human body. So all these capabilities, acquiring these capabilities, it follows a stepwise manner, a sequential fashion. First it has got some immortality basics, right? That mutation it has got. Now it is potential to develop other mutations, so that each mutation will impart a special characteristic to that cell, so that over a period of time, this single cell, it will develop into a group of cells which have got all the potential characteristics of a cancer. Okay, all the characters are present, right? So this is what we call it as natural progression of the cancer. Okay, so if you see here, the genetic damage that is happening here, usually in a normal cell, if it encounters a genetic damage, what happens usually is that cell will be deleted from our system, either by inducing apoptosis, or our immune system will attack that and it will destroy that cell, if at all there is a DNA damage. But cancer cells, the first and foremost character that they develop is non-lethal genetic damage. So the genetic damage that has occurred is just enough to transform the normal cell into a cancer cell, but it is not killing that cell. So it allows the survival of that cell. That's what we call it as non-lethal genetic damage. Because of that, the cell can survive even with the genetic damage. So it will expand, it will go on proliferating and dividing, leading to clonal expansion of the mass. Okay, right. And one interesting fact here is, remember, for a cancer cell, our body is always a foreign body. Okay, it is like it is like an hostile environment. Okay, it cannot usually survive unless it has got that very strong willpower, and it has got that it has to grow in a way that it can survive in a hostile environment. Okay, here comes the Darwinian concept. During this tumor progression, when it is acquiring several mutations, what happens is there will be production of some cells which are inferior to the other cells. So what happens is these inferior cells, they will eventually die in that cancer mass, because they have to uh, fight for their requirements, for their oxygen, for their nutrients, for their protection from immune response. They have to fight with their neighbors. If the next cancer cell sitting in the mass, if it is more potent, oh, definitely that potent cell, it will be selected through Darwinian uh, principle, isn't it? It is survival of the fittest. Whichever cancer cell is the fittest cancer cell, it will survive. Okay, so in this process, what happens is, as the time progresses, an innocent tumor, it will become more and more and more malignant in nature. It will just become like a moron. So, you know one thing, usually when we treat cancer, cancer will be treated by chemotherapy and radiotherapy. We all know that, right? So, once you treat the cancer with certain type of chemotherapy, some amount of development you can see initially. Usually, you will see reducing the size of the cancer, but after some time, they will become even more malignant, so that it can it will stop responding to that chemotherapy itself. That's how a tumor progresses. So in the life cycle of the tumor cancer, there is one initiated cell is there which has suffered a non-lethal genetic damage, so it is expanding clonally. And in this road, in this pathway, it will keep on accumulating several other mutations which impart several phenotypic attributes to the cancer cell, so that it becomes a more aggressive tumor over a period of time. This is what we call it as tumor progression. And at the end of the story, if you look at the cancer mass, to start with, they have started with a set of mutations, but as the tumor is progressing, due to Darwinian pressure over the tumor survival, only the fittest cells, only the cells that have got different kinds of mutation, only they will survive. So the cells, they will become heterogeneous in nature. Of course, they will have all the initial mutations in them, but in addition to that, there will be several other mutations that are added. For example, let us see this picture. For example, this is a normal cell. Okay, this is normal, physiologically normal cell. First, it will acquire that non-lethal genetic damage, so that it escapes the genetic surveillance and it is still surviving. Okay, and it will keep on proliferating, so that it forms a clone, right? So that we call so it has got that dividing capacity, so it is a stem cell like cell, isn't it? So this is a precursor stem cell like cell, and after some time it will start acquiring some of the phenotypic attributes of the cancer. Then we call it as mutate or phenotype. And over a period of time, these things, they will accumulate, these mutations, they will accumulate, and they will transform this normal cell into cancer cell in a sequential manner. See, as the tumor progresses, this cancer cell, it will develop into a mass of cells which have got several which are actually heterogeneous in nature, and they have several other mutations also in them. Okay, so this is how a tumor progresses. This is what we call it as natural history of cancer. So how does this normal cell is transforming into a cancer cell? We are trying to understand the genetic basis of the cancer cell, isn't it? In a normal cell, usually there are 4 sets of genes are present in a physiologically normal cell for its survival, for its growth, for its cell cycle purposes, and to decide when it has to die. To overview the normal physiological functions of a cell, there are 4 sets of genes are there. Okay, these 4 sets, they carry a specific function. They are proto-oncogenes. The first category is proto-oncogenes. These proto-oncogenes are concerned with growth promotion of a cell. The second category is tumor suppressor genes. These genes are the function of these genes is to inhibit the cell growth. So under the influence of proto-oncogenes, a cell will grow. It will divide and whatsoever. Okay. So once the growth is enough, tumor suppressor genes, they will kick in and they will send a signal to the cell so that the cell growth is inhibited. So it maintains a perfect balance between proliferation and suppression of that proliferation. Okay. And the third set of genes is DNA repair genes. The function of these genes is to repair DNA. During cell cycle, there is division of DNA also, isn't it? Apart from cell component synthesis, there is division of DNA material is also there. During this cell cycle progression, there is every chance that this DNA is misread at some point of time, and so a defective gene might be passed on to the daughter generation. So the function of this DNA repair genes is to scrutinize that process, to see, to check the spellings of the DNA so that only correct DNA is passed over to the next generation. Okay, sometimes it will go in a nice fashion, DNA repair is possible, and so we can continue the cell cycle. Or sometimes this repair is beyond repair, this defect is beyond repair. So what happens then is apoptotic genes, this is a 4th set of genes, they will come into play. Whenever they identify a cell with a defective genetic material, they will induce apoptotic genes in that cell so that cell commits self-suicide. Thereby, so what happens, a cell which is carrying a defective gene, it is now undergoing death. So there is no fear of transmitting the defective DNA into the next generation, isn't it? So this is how 4 sets of regulatory genes, they lead to normal physiological growth of the cell. Among these things, we are now going to discuss about proto-oncogenes for today's class. Okay, the other sets of genes will be dealt under separate classes. Okay. So proto-oncogenes and oncogenes. Proto-oncogenes are concerned with growth of the cell, right? How does these proto-oncogenes, they do work usually? They work at different levels. Do you know the sequence of growth factors and receptors? Just look at them. If a cell has to divide, what usually happens is there will be some growth factor which is concerned with the giving positive stimulus for that cell so that it undergoes division, right? This growth factor will come and first bind to a receptor on the cell surface. Okay, and now this receptor will be activated. Till that time it will be in inactive stage. Till the growth factor comes, it will be in inactive stage. Once the growth factor interacts with the receptor will get activated. Okay, once the receptor is activated, it will induce secondary signal molecules in the cytoplasm. These secondary signal molecules, they will be in 2 steps. Okay, we call them as signal transducing molecules. Primary signal transducing molecules will be there, and these primary molecules, they will activate secondary molecules of signal transduction. Okay, and as a result of that, the signals, they will reach the nucleus and they will cause initiation of DNA transcription. Okay, now for a cell to divide, not only the DNA but also the other cell components like cytoplasm, cell organelles like endoplasmic reticulum, ribosomes, everything has to be doubled, right? So for that, other transcription factors are needed. Okay, and cell cycle will now progress and cell will undergo division. This is what usually happens in a normal cell. Okay, so proto-oncogenes, what is the function of these proto-oncogenes is they will act like growth factors. They will act like growth factor receptors and signal transduction molecules and transcription factors and cell cycle components. Under these 5 levels they do act. Okay, see, proto-oncogenes, they encode for growth factors. They encode for growth factor receptors. Look at this. Growth factor receptors. They encode for signal transduction molecules. They encode for transcription factors and they also encode for other cell cycle components. This is how a proto-oncogene works. If at all there is a mutation in any one of these levels, the resultant product we call it as oncogene. Try to get familiar with this terminology. Oncogenes are nothing but mutated counterparts of proto-oncogenes. Okay, these proto-oncogenes, the product of these proto-oncogenes are called as oncoproteins. Proto-oncogene products are called proto-oncoproteins. Oncogene products are called oncoproteins. Okay, so the function of these oncoproteins is, these are the genes that are responsible for autonomous cell growth, which is the characteristic feature of cancer cell. Cancer cells, they do not need growth promoting signals. That is how they differ from normal cell. For a normal cell growth, it needs a proper growth factor and proper growth factor sequence. But for a cancer cell, there is no need for such growth promoting signals. Okay, and these cancer cells, they are so crazy that they are even insensitive to the inhibitory signals. Okay, so let us see few examples here. So we are talking about oncogenes and their products, that is oncoproteins. If an oncogene is encoding for a growth factor, what will happen? How does it act? Few tumor cells, certain kinds of tumors, they secrete their own growth factors. For example, gliomas and sarcomas. In case of gliomas, they tend to secrete a growth factor called as platelet derived growth factor. And in case of sarcomas, they secrete a growth factor called as transforming growth factor alpha and epidermal growth factor receptor. So these growth factors, they are produced by the cancer cell itself. Cancer cell is not depending on an external source for the production of their growth factor. So once there is continuous supply of the growth factor, what will happen to the cell fate? It will go on dividing, isn't it? So that's how a growth factor which is encoded by a oncogene will lead to cancer progression. Okay, and the second thing is growth factor receptors. Certain kinds of tumors, they have got a receptor which is continuously in a active stage. We have seen in a normal cell, unless the growth factor comes and bind to the receptor will be sleeping. Only after growth factor comes and binds to that, this receptor will be activated and the secondary signal molecules will be activated, isn't it? But in case of a cancer cell, these growth factor receptors are continuously active 24/7. Okay, all the time they are active. Even they don't need even the growth factor itself for their activation. This we call them as growth factor independent receptors. Okay, so what will happen? Just imagine there is no need for growth factor and these growth factor receptors are continuously active. What will happen to the cell? It will keep on dividing. The best example for this kind of mutations is lung adenocarcinomas and carcinoma breast. Okay, and let's see about signal transduction proteins. I told you once the growth factor receptor is activated, it will activate the secondary molecules in the cytoplasm. primary molecules and secondary signal transduction molecules will be there. Two sets are there, right? So these two sets, sometimes what will happen due to mutations, these proto-oncogenes, they will be converted into oncogenes, which will produce continuously active signal transduction molecules. Okay, the most important signal transduction molecules in tyrosine kinase pathway are RAS. RAS and secondary molecules are MAP kinase pathway and PI3K pathway. So these are all, we can group them together as signal transduction molecules. Once there are mutations that are affecting this signal transduction molecules, they are constituently active, of course, right? So what will happen? Cell will continuously receive a signal to further proliferate and further proliferate. So the cell cycle, it keeps on going. So the best examples for this type of cancers are pancreatic adenocarcinoma, cholangiocarcinoma, CA colon and carcinoma endometrium, thyroid cancers. In all these cancers, the signal transduction molecule that undergoes mutation is RAS. Okay, and there is one more signal transduction molecule called as RAF. Okay, this is mutated in hairy cell leukemia. Okay, right. So if you observe, these oncogenes, they are acting at different levels. One is at a growth factor level. There is continuously production of growth factor, and one thing at a growth factor receptor level, right? Constituently active growth factor receptors. And the third level is signal transduction levels. So the signal transduction molecules are mutated here, so it is leading to proliferation of the cell ultimately. Okay, so these proto-oncogenes which are converted into oncogenes, they act at several different levels: growth factor level, growth factor receptor level and signal transduction level. Okay, and one very important oncogene, in fact it is a proto-oncogene which needs worth mention is, so this MYC gene is located on chromosome number 8 in all eukaryotic cells including human beings. Okay, and MYC gene is a very important gene which has got several regulatory functions in cell cycle. Okay, it decides whether the cell cycle has to progress or not. Okay, and it is very much important for protein synthesis. Several protein synthesis is under the control of MYC gene, and it has got a special property, very special property, that it can reprogram a somatic cell into pluripotent stem cell. Okay, it can virtually reverse all the fate of a cell, and it can transform a normal somatic cell into a cell that has got abundant proliferative potential like a pluripotent stem cell. Okay, so it is, you can now easily understand this. If only a MYC gene is altered, so the cell will definitely lose control over its cell cycle proliferation and its protein synthesis, and it might impart a pluripotent stem cell like properties, isn't it? So MYC gene is one very important example for proto-oncogene. Okay, the oncogenic versions of MYC gene are usually found in Burkitt lymphoma and neuroblastoma. Okay, so this is about proto-oncogenes and oncogenes. Just remember, oncogenes are nothing but mutated counterparts of proto-oncogenes. Proto-oncogenes are concerned with cell proliferation and cell growth regulation. If cell proliferation and growth regulations are altered, cell suffers from high proliferation rate, which is one of the characteristic of cancer cells. Okay, so the second set of genes that we will be discussing today is tumor suppressor genes. Okay, it is very easy to remember. Tumor suppressor genes, the function of them is they usually control cell proliferation. Tumor suppressor genes are the normal genes. Okay, and the function of them is to control cell proliferation. They will suppress the unwanted cell proliferation. Okay, so just imagine if these tumor suppressor genes are not functioning properly. In other words, if there is loss of function, what will happen to the cell growth? It leads to uninhibited cell proliferation, isn't it? Okay, and tumor suppressor genes, once they are damaged, it also leads to genetic instability of the cell. it will make the cell more vulnerable to acquire so many other mutations. So it becomes genetically unstable, okay, which is again a characteristic feature of a cancer cell, okay. But for a tumor suppressor gene knockout effect, for it to established on a full scale, both the alleles must be mutated. Both the alleles must be mutated in a tumor suppressor gene so that cell will lose control over proliferation, okay. And the best examples for tumor suppressor genes are APC gene, okay, which is mutated in colonic adenocarcinoma. RB gene, you will be hearing these two genes throughout your general pathology, RB gene and TP53 genes. These two genes are very important tumor suppressor genes. RB gene, also called as retinoblastoma gene, it is found, the mutated part is found in retinoblastoma and osteosarcoma. TP53 is mutated in Li-Fraumeni syndrome. WT1 gene is also called as Wilms tumor gene 1. So as the name says, it is mutated in Wilms tumor. And we have got BRCA1 and BRCA2 genes also, which are mutated in carcinoma breast. So these are all the tumor suppressor genes and which are mutated leading to carcinomas. So with this, we conclude today's topic of proto-oncogenes and oncogenes and tumor suppressor genes. Okay, thank you so much.